Hello students, I am Dr. Shakila welcoming you to E-Pathshala. Today I will be speaking about principles and theory and factors involved in encapsulation. Encapsulation is defined as a technology of packaging solids, liquids or gaseous materials in miniature sealed capsules that can release their contents at controlled rates under specific conditions. Encapsulation in food processing includes the coating of minute particles of ingredients, example acidulants, fats and flavors, as well as whole ingredients, example resins, nuts and confectionery products, which may be accomplished by microencapsulation and macrotechnic coating techniques respectively. The microcapsule has the ability to preserve a substance in the finely divided state and to release it as occasion demands. Criteria to select a proper encapsulation technology. Ideally, the choice for a certain encapsulation technology should not be based on trial and error. The desired properties might be based on consumer insights, nutritional demands or ideas to improve food processing and storage stability. Consumers also debate on whether encapsulation is the right technology to bring the desired benefit. When one chooses encapsulation as a technology to deliver the desired benefit, one should consider carefully the design of the encapsulate regarding the following aspects: physiochemical characteristics of the active material, the processing conditions that are used during food production or processing, storage of the encapsulates prior to use, the storage conditions of the food product containing the encapsulates prior to consumer use, particle size and density needed to have it incorporated properly in the food product, the trigger and mechanism of release, and finally the cost constraints. Based on this analysis, one should also consider selection of the coating, process of encapsulation to be followed, legal issues to be considered, preparation at sufficient quantities, constant quality, and at the right time to ensure proper supply chain. In the food industry, encapsulation process can be applied for a variety of reasons. Encapsulation is a useful tool to improve delivery of bioactive molecules, example antioxidants, minerals, vitamins, phytosterols, lutein, fatty acids, lycopene, and living cells such as probiotics into foods. In most cases, encapsulation refers to a technology in which the bioactive components are completely enveloped, covered and protected by a physical barrier without any protrusion of the bioactive components. These are later released at controlled rates over prolonged periods and under specific conditions. Encapsulation provides an effective method to cover an active compound with a protective wall material and thus offers numerous advantages. The bioactive components include lipids, vitamins, peptides, fatty acids, antioxidants, minerals and living cells such as probiotics. Some of the main benefits are protection of various actives against evaporation, chemical reactions or migration in food, controlled delivery and preservation of the stability of the bioactive compounds during processing and storage, prevention of undesirable interactions with other components in food products, and masking unpleasant feelings during eating. Encapsulation is an important approach to meet all demands by delivering the bioactive food components at the right time and right place. An attractive possibility is to use a methodology where two or more bioactive components can be combined to have a synergistic effect. With advances strategies for stabilization of food ingredients and development of new approaches, we will be able to improve nutritional properties and health benefits of food compounds. Encapsulation also constitutes a competitive tool to create new properties, usual materials. Getting stable functional ingredients is one thing, but it is not enough if we cannot integrate them easily in the food. Many vitamins, plant extracts, unsaturated fatty acids are hydrophobic liquids. Mixing such compounds in a hydrophobic food powder can prove to be a real challenge. In addition to protection, microencapsulation allows the conversion of ingredients to free-flowing easily suspendable and dispersible powders. Unsaturated fatty acids are recognized as beneficial for health. However, they sometimes have an unpleasant taste which can become unacceptable when they are oxidized. Encapsulation largely overcomes this problem by taste masking and limiting oxidation. Additionally, incorporating flavors in the coating helps to make the functional food pleasant to consume. Encapsulation can also be used as a tool for innovation. For example, the company Orbitz in Canada sells a drink containing a suspension of colored capsules containing different aromas and or some vitamins, thus making functional food consumption a fun experience. The company Salvona has developed encapsulation technologies allowing sequential release of aromas and sensory ingredients in functional foods. Encapsulation can also be used to enclose solids, liquids or gases inside a micrometric wall made of hard or soft soluble film, forming small particles called microcapsules. The polymer acts as a protective film, isolating the core and avoiding the effect of its inadequate exposure. This membrane dissolves itself through a specific stimulus, releasing the core in the ideal place or at the ideal time. Microcapsules offer food processors a mean with which to protect sensitive food components, ensure against nutritional loss, utilize otherwise sensitive ingredients, incorporate unusual or time release mechanisms into the formulation, mask or preserve flavors and aromas, and transform liquids into easily handled solid ingredients. In the following figure, the microencapsulation as well as the encapsulation of foods along with the various examples have been indicated. The food industry applies encapsulation process for a variety of reasons, beginning with encapsulation or entrapment can protect the core material from degradation by reducing its reactivity to its outside environment, example heat, moisture, air and light. Evaporation or transfer rate of the core material to the outside environment is decreased or retarded. The physical characteristics of the original material can be modified and made easier to handle. The product can be tailored to either release slowly or over a time or at a certain point to control the release of the core material to achieve the property delay until the right stimulus. The flavor of the core material can be masked. The core material can be diluted when only very small amounts are required, yet still achieve a uniform dispersion in the host material. And finally, it can be employed to separate components within a mixture that would otherwise react with one another. Important considerations during the development of encapsulated products include core properties like the chemical structure, solubility and stability, product format such as liquid or powder format depending on final application, particle size including bulk density and color, the amount of bioactive loading in microcapsules, release trigger mechanism, example dissolution, pressure, heat and shear, storage conditions and shelf life requirements such as refrigerated or ambient storage, and finally legal and regulatory requirements for addition into food in the country of its application. From a commercial perspective, there is the additional factor of material and production costs and whether the final product can bear the additional cost of using a microencapsulated ingredient. Explaining the principle, encapsulates are basically particles with a core shell structure, but some of them can have a more complex structure, example in a form of multiple cores embedded in a matrix. Particles have physical, mechanical and structural properties including particle size, size distribution, morphology, surface charge, wall thickness, mechanical strength, glass transition temperature, degree of crystallinity, flowability and permeability. The purpose of encapsulation is often to stabilize an active ingredient, control its release rate and or convert a liquid formulation into solid which is easier to handle. A range of edible materials can be used as shell materials of encapsulates, including polysaccharides, fats, waxes and proteins. The figure shows the different encapsulation layers along with the core material, which is the nutrient. The substance that is encapsulated may be called the core material, the active agent, fill, internal phase or payload phase. The substance that is encapsulating may be called the coating, membrane, shell, carrier material, wall material, capsule, external phase or matrix. Generally, microparticles consist of two components, core material and coat or wall or shell material. The core material implies the material to be coated. It may be liquid or solid or gas. Liquid core may be dissolved or dispersed material. Two main types of encapsulates might be distinguished, that is, the reservoir type and the matrix type as shown below. The reservoir type has a shell around the active agent. This type is also called capsule, single core, monocore or core shell type. Application of pressure can lead to breakage of the reservoir type of encapsulates and thus to the release of its contents. The active agent in the matrix type is much more dispersed over the carrier material. It can be in the form of relatively small droplets or more homogeneously distributed over the encapsulate. Coating material. This is an inert substance which coats on core with desired thickness such as inert polymer, plasticizer, coloring agent, resins or waxes and lipids. Let's look at the properties of an ideal coating material. The coating material should possess good rheological properties at variable concentrations, allowing for easy for manipulation during the process of encapsulation. It should possess the ability to disperse or emulsify the active material and stabilize the emulsion produced. It should not react with the material to be encapsulated both during processing and on prolonged storage. Should be able to seal and hold the active material within its structure during processing or in storage. The coating material must provide maximum protection to the active material against environmental conditions such as heat, light and humidity. It should be soluble in solvents acceptable in the food industry, example water, ethanol, etc. It should meet specified or desired capsule solubility properties and active material release properties. Economy of food grade substance. And finally, approved by a controlling authority. Generally, capsules can be classified according to size. In terms of their shape and construction, capsules can be divided into two groups, microcapsules and microspheres. Microcapsules are particles consisting of an inner core almost placed at central containing the active substance which is covered with a polymer layer constituting the capsule membrane. Microcapsules can be classified into three basic categories as monocode, polycode and matrix types as shown below. Monocode microcapsules have a single hollow chamber within the capsule. The polycore microcapsules have a number of different sized chambers within the shell. The matrix type microparticle has the active ingredients integrated within the matrix of the shell material. However, the morphology of the internal structure of a microparticle depends largely on the selected shell materials and the microencapsulation methods that are employed. The most significant feature of microcapsules is their microscopic size that allows for a huge surface area. For example, the total surface area of 1 millimeter of hollow microcapsules having a diameter of 0.1 millimeter has been reported to be about 60 square meter. The total surface area is inversely proportional to the diameter. This large surface area is available for sites of adsorption and desorption, chemical reactions, etc. that may be a part of the encapsulation process. Microspheres are matrix systems in which the core is uniformly dispersed and or dissolved in a polymer network. Microspheres may be homogeneous or heterogeneous depending on whether the core is in the molecular state or in the form of particles which may be suspended respectively. Factors involved in encapsulation techniques. Encapsulation often involves a basic understanding of the general properties of microcapsules, namely the nature of the core and coating materials, the stability and release characteristics of the coated materials, and the microencapsulation methods. The intended physical characters of the encapsulated product and the intended use of the final product must also be considered. Looking at the core material is defined as the specific material to be coated, can be liquid or solid in nature. The composition of the core material can be varied as a liquid core can include dispersed or dissolved material. The solid core can be a mixture of active constituents, stabilizers, diluents, and the release rate retardants or accelerators. Looking at the coating materials, it should be capable of forming a film that is cohesive with the core materials, be chemically compatible and non-reactive with the core material, and provide the desired coating properties such as strength, flexibility, impermeability, optical properties, and stability. The total thickness of the coatings achieved with microencapsulation techniques is microscopic in size. Stability, release, and other properties. Three important areas of current microencapsulation application are the stabilization of core materials, the control of release or availability of core materials, and separation of chemically reactive ingredients within a powder mixture. A wide variety of mechanisms is available to release encapsulated core materials, such as disruption of the coating can occur by pressure, shear, or abrasion forces, permeability changes brought about enzymatically, etc. Improved gastrotolerability of drugs can be obtained by microencapsulation. Physical character of the final product. Microcapsules should have desirable physical properties like ability to flow, to be compacted or to be suspended, and the capsule wall must be capable of resisting the pressure during compression, etc. Finally, the coating materials. A number of different substances, both biodegradable as well as non-biodegradable, have been investigated for the preparation of microcapsules. These materials include the polymers of natural and synthetic origin and also modified natural substances. Factors involved in encapsulation. One of the most important reasons for encapsulation of active ingredients is to provide improved stability in final products and during processing. For example, probiotics are highly sensitive to variation of pH, mechanical stress, transport conditions, and digestive enzymes in the stomach. At present, probiotics are the driving force in the design of functional foods, especially in dairy products, maintaining their functional effects for supporting human health. These living cells need to survive the food process, storage, and food intake before they can be useful. Encapsulation increases not only their bioavailability but more importantly functionality. A choice of an encapsulation system is always crucial since it has to be efficient and easily incorporated into the food without interfering with the texture and taste of the food. Another factor involved in the encapsulation process is the decrease in evaporation and degradation of volatile actives such as aroma, which usually contains a mixture of volatile and odorous organic molecules. As flavors are usually expensive, food manufacturers are usually concerned about the preservation of aromatic additives which needs to be preserved. Aroma component is usually encapsulated with a protective wall material against evaporation, chemical reactions, or migration in a food. Since flavor is one of the most important characteristics of food, the ultimate goal of encapsulation is to control aroma release and to improve stability during processing and consumption of the final product. In general, aroma releases from food before and after eating, which depends on the aroma features and physical state of the matrix. The success of an encapsulation process is often linked to know-how of the formulation or of the chemistry to achieve stabilization. This is especially the case in the food industry, where the number of acceptable materials is very limited. In most cases, researchers are essentially concerned with finding the optimum formulation matching with a specific method. The engineering aspects are often neglected, while probably a substantial part of success to maintain the integrity of active compounds, provide the right properties to microcapsules, and reduce the cost is directly related to the designing and operating parameters. Finally, to conclude, advances in the development of microencapsulation technology for food applications have been driven by the need for the core to be encapsulated, new and alternative materials that are cost-effective encapsulants, and materials which will withstand the processes widely used in the food industry. Understanding the core, the final application, and the mechanism required to release the core is essential for effective design of microcapsules release characteristics. Different release characteristics can be achieved depending on the requirement, example, controlled, sustained, or delayed release. The actual production of encapsulated food ingredients represents thousands of tons per year and is estimated to be growing at around 10% annually. However, many challenges still remain. For example, incorporation of water sensitive ingredients in high moisture foods is not solved yet, because most microcapsules impermeable to water are not soft enough and will be detected by the consumers. More recent developments in encapsulation technologies for food applications have focused on applying the technology to more cost-effective food grade encapsulant materials and process available in the food industry. The need for controlled release and delivery of bioactive food ingredients to target sites in the body continues to drive other new developments. Converting stable microcapsule formulations into powders is still the preferred option for production of microencapsulated bioactive ingredients, as it offers more convenience and flexibility. An understanding of how these formulations will behave during the drying process and on reconstitution is critical to the success of powdered preparations. There is also a growing need to find suitable solutions that provide high productivity and at the same time satisfy an adequate quality of final food products. Future researchers should be focused on overcoming the gap between conditions at research level and demands for large-scale applications, improving existing manufacturing technologies, choosing new processing conditions and new carrier materials. Also, future studies should be oriented to preservation and storage techniques that could be easily adopted at the industrial level. Developing an encapsulated product remains a challenge, requiring a multidisciplinary and integrated approach. The key for success most often goes through the support and the expertise of specialized companies. Many encapsulation technologies currently exist, but many of them are still at the development stage. Once fully tested and validated, these new methods will undoubtedly broaden the spectrum of possible applications for this versatile technology.